Now, when most students encounter biochemical pathways, this is probably what enters their mind. Our goal in this module and the series of modules in biochemistry is to simplify things and make sure that you'll be able to comprehend, understand the must-know concepts, and correlate it with other clinical subjects. Now, let's talk about the electron transport chain. This is also known as the respiratory chain. So the other name of the respiratory chain is the electron transport chain, or the ETC. Now, what should we know about the electron transport chain? Number one, it is considered to be the final common pathway in all aerobic cells by which electrons, which are actually derived from various substrates and various peripheral pathways, are transferred to oxygen. So in the electron transport chain, electrons are going to be passed from one complex to another to combine with oxygen to form water. Now, the pearls you have to remember regarding the electron transport chain is it is the final common pathway in aerobic cells. Now, this pathway occurs in the inner mitochondrial membrane, and the final acceptor of the electron transport chain is oxygen. There are several complexes in the electron transport chain, which we will discuss, and electrons are passed from one complex or member of the electron transport chain to another until it combines with oxygen, which is the final acceptor. So, other pearls you have to remember regarding the electron transport chain is, where does this important pathway occur? This pathway is located in the inner mitochondrial membrane. And this is where a very important biochemical reaction occurs. And this is what we call oxidative phosphorylation. Okay, again, oxidative phosphorylation. This is the production of ATP, which is coupled to the electron transport chain. Now, theoretically, there are 4 complexes, but later we will mention that there's actually a 5th complex. So these components of the electron transport chain are organized into 4 complexes whose role is to accept and transfer electrons until you reach the final acceptor, which is oxygen. Now, in this photo, okay, let's try to dissect this. In this photo, what you will see are 4 complexes. This occurs in the inner mitochondrial membrane. So again, in the inner mitochondrial membrane. So what I want you to remember is that there are 4 complexes. So what are these 4 complexes? So we have complex 1, complex 2, complex 3, and complex 4. Now you will notice the hydrogens there, the letter H. So there's basically the transport of one electron from one complex to the other complex. Now here, as you can see, you have the inner mitochondrial membrane. And inside the inner mitochondrial membrane is a very important process, which is called oxidative phosphorylation. But before we mention oxidative phosphorylation, I want to mention another form of phosphorylation, or cellular production of energy. which occurs outside of the mitochondria. And this is what we call substrate level phosphorylation. Okay, again, this is what we call substrate level phosphorylation. Now, let's try to isolate this. Okay, so again, there is substrate level phosphorylation, which is defined as the synthesis of ATP from ADP. So it's the synthesis of adenosine triphosphate from adenosine diphosphate. Now, the classic example here is your phosphoenol pyruvate being converted to pyruvate. Now, this is actually a very important step because this is the last step of glycolysis, wherein PEP, which is a high energy compound, will be converted into pyruvate. Now, in complete definition, substrate level phosphorylation is the synthesis of ATP from ADP by phosphorylation, which is coupled to the exergonic breakdown of a high energy compound. In this case, the high energy compound is phosphoenol pyruvate, or PEP. So this is the step before pyruvate is formed. This is the last step of glycolysis. Now, I want everyone to memorize these 3 enzymes in the yellow boxes. This is what is found in our Harper's Biochemistry. 3 classic examples of substrate level phosphorylation. So again, how did we define substrate level phosphorylation? We said this is the production of ATP from adenosine diphosphate, which involves phosphorylation, which is coupled by the exergonic breakdown of a high energy compound. These are the 3 examples of substrate level phosphorylation in our textbook. Number 1, the one in the middle, involves pyruvate kinase. So this is the reaction wherein phosphoenol pyruvate is converted to pyruvate. And as you can see, ADP will be converted to adenosine triphosphate. Remember, the energy currency of the cell is ATP. That's why ADP, which is adenosine diphosphate, has to undergo phosphorylation. So dagdagan ng phosphate, you add a phosphate. So the diphosphate becomes a triphosphate. The other 2 examples of substrate phosphorylation would involve phosphoglycerate kinase and succinyl-CoA synthase. So memorize 3 enzymes involved in substrate phosphorylation. Put pyruvate kinase number 1 on the list, phosphoglycerate kinase, and lastly, succinyl-CoA synthase. Now, what about oxidative phosphorylation? This is another method or means wherein there's cellular production of energy. In oxidative phosphorylation, ADP is phosphorylated. So there's phosphorylation of adenosine diphosphate. However, this process is coupled with the oxidation of a substrate, which is linked to the electron transport chain. So how do you differentiate substrate level versus oxidative phosphorylation? In substrate level phosphorylation, ADP is phosphorylated, but this is coupled to the breakdown, exergonic breakdown of a high energy compound. And what about oxidative phosphorylation? The phosphorylation of ADP is coupled to the electron transport chain. Now, this is always asked in exams. Where is the site of oxidative phosphorylation. In what organelle? In what cellular organelle? Where does oxidative phosphorylation occur? It occurs in the inner mitochondrial membrane. Now, there is a very important theory, a hypothesis, which is known as the chemiosmotic coupling hypothesis of Peter Mitchell. This is known as Mitchell's chemiosmotic coupling hypothesis. Okay, again, the chemiosmotic coupling hypothesis of Peter Mitchell. And this hypothesis states that there is a tight coupling of the transfer of electrons and phosphorylation. So there is a tight coupling of electron transfer, which is oxidation, and phosphorylation. So this hypothesis is known as the chemiosmotic coupling theory. So flashcard to remind everyone, where does oxidative phosphorylation occur? It occurs in the inner mitochondrial membrane. Okay, it occurs in the inner mitochondrial membrane. Now, earlier we mentioned there are 4 organized complexes in the electron transport chain. Complex 1, 2, 3, and 4. It is mandatory for you to memorize each complex if you are heading to the exams. So please memorize complex 1. This is NADH dehydrogenase. Complex 2, this is succinate dehydrogenase. Complex 3, this is cytochrome complex. And of course, complex 4 is cytochrome oxidase. Now, please write down that complex 5 is ATP synthase. Complex 5 is ATP synthase or ATP synthetase. So I hope you got this. NADH dehydrogenase, this is complex 1. Succinate dehydrogenase is complex 2. Cytochrome complex is complex 3. And cytochrome oxidase is complex 4. Complex 5 is ATP synthase. So here is a lifted diagram showing you the different complexes inside the inner mitochondrial membrane. Now here is also showing you a representation of the enzyme complexes within the inner mitochondrial membrane. Now here on your right, your far right, is ATP synthase. This is complex 5. Okay, this is complex 5, ATP synthase. Now, this is what's going to come out in any standard exam in biochemistry, and the topic will touch the electron transport chain. This is site-specific inhibition of the electron transport chain. There are certain substances which will specifically inhibit either complex 1, 2, 3, or 4, or complex 5. So let's start the ball rolling. So in the first column, you have the complex, the components of the electron transport chain, the name of the enzyme, and the inhibitors. Now, please pay attention to the enzymes, the inhibitors, the substances which are highlighted in red. These are previous board recalls. So complex 1, which is NADH, is inhibited by barbiturates, amytal, rotenone, and pyridine A. So emphasis on barbiturates, amytal, rotenone. For complex 2, which is succinate, this is inhibited by pesticides like carboxine, malonate, and TTFA. I will explain to you in a while what TTFA is. So examples of barbiturates would be pentobarbital, phenobarbital, secobarbital. So just examples of barbiturates: pento, seco, phenobarbital. So barbiturates, amytal, and rotenone will inhibit what complex? Complex 1. What inhibits complex 2? Carboxine, malonate, and TTFA. Please take note of this. Now, what is TTFA? This stands for phenyl trifluoroacetone. This is a chelating agent which inhibits specifically complex 2. So again, what is TTFA? Phenyl trifluoroacetone. So TTFA. Now, let me introduce the term uncouplers. So what are uncouplers? Uncouplers are compounds or substances that will allow oxidation to continue without corresponding synthesis of ATP. Again, uncouplers are compounds that allow oxidation to continue without corresponding ATP synthesis. Now, take note of these examples in the book. What is highlighted in red, your thermogenine, this is what came out in the recent boards. So we have 2,4-DNP or 2,4-dinitrophenol, aspirin whenever in excess, and thermogenine. Thermogenine. So please take note of this. So uncouplers, compounds that allow oxidation to continue without corresponding ATP synthesis, okay? Special emphasis on 2,4-dinitrophenol and your thermogenine. Now let's isolate thermogenine. This is also known as your uncoupling protein 1 or UCP1. Uncoupling protein 1. Now thermogenine is actually found in the mitochondria of your brown adipose tissue. You usually see this in newborns. And one of the functions of thermogenine is to help generate heat by non-shivering thermogenesis. So thermogenine is found in the mitochondria of brown adipose tissue. So as the word indicates, the prefix thermo, which means heat, okay? So it's used to generate heat, most especially in newborns. So thermogenine is an uncoupler. Now, I want everyone to memorize oligomycin. Okay, This is a non-specific inhibitor of the electron transport chain. Oligomycin will block the FO fraction of your enzyme ATPase. ATPase is the same enzyme we mentioned earlier, ATP synthase. So ATPase the enzyme has subunits or fractions. Specifically, the FO fraction is inhibited by oligomycin. So oligomycin as a big letter O blocks what fraction? FO. So FO is oligomycin. Now, what happens if you block the enzyme ATPase? It's simple. ATP will not be produced. If you remember, this is complex 5 of the electron transport chain. So let's go back. What blocks complex 1? Rotenone barbiturates, amytal. What blocks complex 2? TTFA. And what do we call those compounds that allows oxidation to occur? However, there is no synthesis of ATP. We call them uncouplers. And what are the uncouplers I want you to memorize? Number 1, thermogenin. Number 2, 2,4-dinitrophenol. Now, a non-specific inhibitor is oligomycin. And oligomycin blocks the FO fraction of the enzyme ATPase. So here, let me discuss this. 2,4-dinitrophenol has a unique mechanism because it will actually alter or destroy the pH gradient along the membrane. So this is the mechanism by which it serves as an uncoupler. Now, always remember your chemiosmotic coupling theory, wherein oxidation is tightly coupled with phosphorylation. What is the moving force for electrons to be transported from one complex to another? It is the electrochemical gradient. And this electrochemical gradient is affected by the pH. So if you destroy the pH gradient, you are actually preventing ATP from being synthesized. So here, uncouplers of oxidative phosphorylation, 2,4-dinitrophenol or 2,4-DNP. Now, look at this visual mnemonics. What are the last 3 letters of the drug rotenone? It's O-N-E, spelled as 1. So what complex does rotenone block? It's complex 1, because the last 3 letters of rotenone is 1. Now, other drugs or compounds that would inhibit complex 1, barbiturates. Okay, I'm just going through a few flashcards to make sure everyone gets this before we're about to finish this module. So complex 1 is barbiturates. Now, what about complex 3, 4, and 5? So complex 3 is cytochrome complex. The inhibitors would be antimycin A and dimercaprol. Cytochrome oxidase, this is complex 4. The inhibitors is cyanide, sodium azide, hydrogen sulfide, and carbon monoxide. Now tip, what is highlighted in red? Cyanide and carbon monoxide. These are the ones that recently popped out in the actual boards. Complex 5, which is your ATP synthase complex, is inhibited by oligomycin. So recap, what blocks complex 3? Dimercaprol. What blocks complex 4 or cytochrome oxidase? It's cyanide and carbon monoxide. And lastly, what blocks ATP synthase complex or complex 5 is oligomycin. And remember the specific fraction which oligomycin inhibits, it's the FO fraction. So I'll give you a minute to go over this. So I hope everyone got this. This is very quick and straight to the point. So let's run a few flashcards before we end this module. So what blocks complex 4 of the electron transport chain? It's cyanide. What blocks complex 4 aside from cyanide? It's carbon monoxide. And what blocks complex 5 or your ATPase? It is the drug oligomycin. So that ends our very quick module on the electron transport chain. And those are the must-knows you should bring with you to the board exam. Thank you for listening.